Okay, so we're continuing on with the process of transcription. So remember, DNA is in the nucleus, and DNA has all these genes in the old. They used to think that human DNA had 100,000 genes, but after the Human Genome Project, they realized that no, human DNA actually holds about 20 to 25,000 genes, and each gene can actually be used to make different proteins and different parts of different polypeptides. Um, Within each gene, there are things called exons and introns. The exons are segments of the gene that actually code for protein, and the introns, they don't code for anything. Okay, so in the red boxes, we have the introns, all right? Um, so the first RNA that is transcribed, which is that heteronuclear or heterogeneous nuclear RNA, hnRNA, just transcribes the whole thing, the exons and the introns. So the things that are not in a red box, those are the exons. The things in a red box, those are the introns. What you have to do is get rid of the introns in the RNA before you send it out to the ribosomes to get translated and turned into amino acid language. So the snRNA, the small nuclear RNA, is what helps splice out those introns. So what we're going to do is we're going to just indicate where the introns, the copy of the introns, the complementary copy of the introns are. And then your snRNA is going to come and first this is going to go away. It's going to splice this away and splice this away. Now, before it becomes actual mRNA, messenger RNA, usually there's something called the GTP cap on the 5' end, and then a poly A tail, lots of AAAAA, adenosine monophosphates are added in at the 3' end. But the most important thing in processing this RNA is getting rid of the introns. Okay, once that is done, and now the mRNA is ready to move over to the cytoplasm to get read at the ribosomes and get translated by the tRNA. Now, what happens to the template strand and the informational strand of the DNA? They just come back, recoil, form a double strand, and they're ready to be transcribed again when needed. Okay, now we're going to use something called the codon table, and the codon table is in your book, and you can look it up and make a copy of it, print it out, just have it with you. But that codon table is read 5', 3'. So these exons here, we're going to be dividing them up into triplets or codons, but we have to do it 5', 3'. Notice this complementary strand that we created is 3', 5'. So we need to flip it, all right? So this is what I have to do right now, is take this strand and flip it, 5'. So I'm going to write G, G, C. I'm omitting the intron, the copy of the intron, because remember we spliced it away. A, 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 C, A, 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 U. 3'. Flipped it over because, like I said, the codon table is read 5', 3'. Now, remember how I said put a GTP cap here, poly A tail here. We got rid of the introns. Now we can call this mRNA or messenger RNA. All right, so I can get rid of all of this now. Now I have my messenger RNA. And where does it go? It goes to the ribosomes. Again, if you've had a bio class, you know that ribosomes are in the cytoplasm. And this is where protein synthesis occurs. Now, ribosomes are made up of these particles. They have a big subunit and a small subunit. And the ribosomes are made up of rRNA, ribosomal RNA, and protein. So we left the nucleus, and now we're going to the ribosomes. And the ribosomes are made up of rRNA, which is ribosomal RNA, and protein. And if you look at little diagrams of the ribosomes, they have the big subunit and the small subunit. Big subunit, small subunit, big subunit, small subunit. Now this mRNA is going to go through these ribosomes, or you can think of the ribosomes allowing the mRNA to go through them. I always think of it as a bunch of needles, and the mRNA is the thread going through the holes of all the needles, all right? The mRNA is going to be divided up into codons, the triplets. So each 3 are a codon. Each 3 are a codon. Each 3 are a codon. Each 3 are. So we've got 3, so 1, 2, 3, 4 codons. So this hypothetical peptide of ours is going to be coding for 4 amino acids, 4 amino acids. Each codon, this is sometimes called a triplet, but the codons are here in the mRNA. Now, we're done with transcription and we're ready to do translation. Translation. Now, translation occurs in the cytoplasm, it occurs at the ribosomes, and this is where nucleotide language is now translated into amino acid language. There are these RNA molecules called tRNA. tRNA, this is transfer RNA. mRNA, remember, was messenger RNA. Transfer RNA sort of looks like this. Okay, there's a 3' end, there's a 5' end, and let me just check on one thing here. Yep. So your amino acid, this is the 3' end, this is the 5' end, your amino acid is right over here. So these tRNAs are carrying amino acids. Any protein that you eat every day in your diet, what is the purpose of eating your protein? To provide your body with a whole bunch of amino acids so that your body can build its own new proteins. So all those amino acids you've just been eating, now each tRNA is carrying a specific amino acid. All right. How does the tRNA know which amino acid to pick up? Well, each tRNA down here, right down here, is something called an anti-codon. Basically, it is complementary base pairs with the codon. So this anti-codon here would be CCG down here, all right? And then the other tRNA, the next tRNA, would have an anti-codon that is UUU, complementary base pairs to this. Now, if you look at the codon table, and it's in your book, so make sure you look at it, and we read the codon table by looking at the first nucleotide, and then the middle nucleotide, and then the third nucleotide in each codon. If you look up GGC in the codon table, you will see that this stands for glycine. So this first tRNA that's at the ribosomes is carrying the amino acid glycine. Okay? Now, look up AAA in the codon table, and you will see that that is lysine. So the next tRNA is going to have lysine there. And as the tRNAs come one after the other, peptide bonds are beginning to be formed between the amino acids. So you end up creating, as they're going through here, glycine, and then it's lysine is attached to it. So, and then another amino acid comes and gets attached to it. So as we go through, your peptide is growing and growing as the peptide bonds are being formed. The next amino acid, according to this, is threonine. I do not expect you to memorize the codon table at all. Make sure you have it with you when you're doing your homework and when you're doing your quiz, because nobody memorizes a codon table. Even though sometimes I used to tease my students in class and say, of course, you have to memorize it, but you don't. And then the fourth one in our hypothetical here is asparagine. All right, and as this is growing, asparagine, peptide bonds are forming, and now you've just created the primary structure of your hypothetical protein polypeptide, okay? Remember, primary structure is just the amino acid sequence. You start to coil it, so secondary structure, and then it starts to fold, and as it gets bigger, it forms tertiary structure, and then you've got a completely perfect protein, proper folding, proper amino acid, proper everything, and now it's ready to be sent out and to do its thing, okay? One error, one small error in one nucleotide could potentially mess up the whole thing. And we'll talk about mutations and how it could affect your protein structure. Okay, all right. I think we have just one more video and we'll be all set with this chapter.